Okay, so we're saying that the major differences that exist between eukaryotic cells and the prokaryotic cells are that one, the nucleus are distinct in the eukaryotic cells. They have a membrane-bound nucleus. And so you could actually point to it and say this is the nucleus of the cell. It is well formed with a nuclear membrane. Whereas for the prokaryote, they do not have a distinct nucleus because it is not in the form of a nucleus. Okay, it comes in the form of a nuclear zone, or what we call the nucleoid. It's an area. It doesn't have a membrane that is covering or surrounding that nucleus. And so you can actually point out to it and say, this is the nucleus of the cell. So instead, it's more like a nucleoid, a nuclear area, the area where you will likely find the nucleus. And so they do not have a nuclear what? Membrane. Their nucleus are not bound by membrane. So the membrane, nuclear membrane are absent in prokaryotes, while they are present in the eukaryote. Two, the eukaryotic cells, they have double membrane cell organelles. That means they have other organelles, other components of the cells that are also membrane bound. That means they have membranes or envelopes that envelope them or cover them or surround them, such as organs such as your chloroplast, your mitochondria, the nucleus, and they also have single membrane bound organelles such as your Golgi apparatus, lysosomes, vacuoles, endoplasmic reticulums. They are all what present. So basically, aside just their nucleus, they also have other membrane-bound what organelles. So they have nucleus that are membrane-bound, and then other organelles in the cell that are also what membrane-bound. Whereas in prokaryotes, you have single membrane cell bodies such as your mesosomes are present, endoplasmic reticulum, plastids, mitochondria, microbodies like lysosome and Golgi bodies are absent. They do not have those other membrane-bound organelles which you have in your eukaryotes, such as your um, uh, mitochondria, Golgi apparatus, or Golgi bodies, and all that. They are not present in the prokaryotes. So basically, you could say that the prokaryotes do not have the membrane-bound organelles as you see in the eukaryotes. Number three is the ribosomes. Both prokaryotes and eukaryotes possess ribosomes, but they have different types of what? Ribosomes. Remember that we said that ribosomes are the sites of protein synthesis. We'll get there later, but for now, their ribosomes in eukaryotic cells, their ribosomes are 80S ribosomes. While the prokaryotic cells, their ribosomes are 70S ribosomes. You know, we have ribosomes that are 80S, and for prokaryotes, they are 70S. The S there is a unit of measurement. It's actually a Svedberg unit that measures or that states the rate of sedimentation. It's not actually based on size, but based on the sedimentation rate. The Svedberg unit of sedimentation rate is for eukaryotic cells, they are 80S, while for prokaryotic cells, they are 70S. Number four, for eukaryotic cells, they have distinct compartments in the cell. That is, the cytoplasm and the what? Nucleus. Whereas in the prokaryotic cells, they do not have what? Compartments. When you say compartments, different, like I said, if those organelles are membrane bound, that means they are separated from each other. They are distinct, they are separate. They have different compartments, pockets of organelles here and there. But in prokaryotic cells, they are all found swimming within the cytoplasm. They do not have compartments where you have different parts and organelles within that cytoplasm. That's what we're trying to say here. For eukaryotic cells, they have distinct compartments within the cells. Each of those organelles have distinct compartments. You can place them around and then make out where they are within the cells. Whereas in prokaryotic cells, they are found more like swimming within the cytoplasm. They are not in compartments because they are not membrane bound. Number five, depending upon the species, the number of chromosomes per nucleus varies from 2 to many. That means for eukaryotic cells, they have 2 or more chromosomes. Depending on the species of the eukaryotic cells, they have 2 or more chromosomes. Whereas in the prokaryotic cells, it only has 1 chromosome per cell. When we talk about the chromosome, talking about their genetic material, their DNA. We're not saying that in eukaryotic cells, their chromosomes they can be 2 or more. Whereas for prokaryotic cells, they only have one chromosome. Number 6, for eukaryotic cells, each of the chromosome is linear with its own end free. Each chromosome is linear, so they have two or more. So each of the chromosomes, they are linear. You know what linear is? Like they are straight. They are linear with its two ends free. The two ends are free. They are straight, linear. Whereas for the chromosomes of that single chromosome in prokaryotic cells, they are mostly circular. That means they are circular. They kind of form a circular structure. They are not single. They are not linear. In a straight form with both ends free. This other case, they are circular, which means their ends are bonded together. The chromosome is circular and remain attached to cell membrane at one point. You see, the ends are connected. They are circular and then they are attached to the cell membrane. So they are not linear that you see, unlike what you see in the eukaryotic cells, but they are circular chromosomes with ends that are connected or bound to the nuclear membrane. Number 7, each chromosome for eukaryotic cells, each chromosome has one linear double-stranded DNA complex with histones. That means their DNA have what we call the histone proteins. Whereas for prokaryotic cells, their chromosome has single double-stranded circular DNA molecule and it is not associated with histones. Histones are proteins. We are not saying that for eukaryotic cells, their chromosomes are linear, double-stranded DNA that are attached to histone proteins. Whereas for prokaryotic cells, they are circular double-stranded DNA molecules without being associated with histone proteins. There are no histone proteins attached to their chromosomes. Number 8, each chromosome has one centromere that divides the chromosome into two arms. For eukaryotic cells, their chromosome has one centromere that divides the chromosome into two arms. Whereas for the chromosomes in the prokaryotic cells, they do not have that centromere. They lack the centromere in the prokaryotic cells. So these are basically the differences that exist between the eukaryotic cells and the prokaryotic cells. The cell structure. The cell structure. We have the plant cells and the animal cells. And their structure, that means their morphological structure, how they are shaped, how they appear, they are not the same. Their structure, their composition, their structure, how the cells appear, their shape, their size, their composition, they are not the same at all. So based on their structure, we'll be looking at the plant cells and the animal cells are different in their structures. And so let's look at what their structures entail. For the plant cells, they have regular outline. The plant cells have regular outline and fixed shape, which may be rectangular, polygonal, or cylindrical. For plant cells, they have a fixed shape. Their cell could be rectangular, it could be polygonal, like a polygon, or it could be like a cylinder. They have fixed shape. They are not irregular. You don't say find them that they are rectangular today and then they are circular tomorrow. No. They have a fixed shape for plant cells. They have a regular outline with fixed shape, which may be rectangular in shape or polygonal or cylindrical. 2, they possess a distinct cell wall. The plant cell possesses a distinct cell wall made up primarily and secondary wall, made up of both primary and secondary wall. That means primary cell wall, the secondary layer, which contains substances such as lignin, pectin, tannin, and cellulose. So you could say that plant cells they have cellulose in their cell wall, cellulose cell wall. They have these, not just cellulose, but tannin, lignin, pectin. These structures, this composition in their cell walls make them rigid and strong. Remember I said they have definite shapes. They are not irregular. So it means that their cell walls are strong. And what makes them that solid, that strong, is because they possess these structures and these components in their cell wall, such as the cellulose, the lignin, the pectin, the tannins, and all that, that makes them distinct cell wall with these layers or walls, both the primary and the secondary walls, making them rigid and strong. Three, the cytoplasm of the plant cells contains endoplasmic reticulum, Golgi apparatus, mitochondria, ribosomes, vacuoles, chloroplast, and nucleus. Within their cytoplasm, within the space surrounded by the cell membrane or the plasma membrane called the cytoplasm, they contain these cell organelles such as your endoplasmic reticulum, Golgi apparatus, mitochondria, ribosomes, vacuoles, and chloroplast, including nucleus. These are some of the structures or the composition or the makeup of the plant cell. They are regular in outline, they have regular shape, they have a fixed shape. It could be rectangular, cylindrical, or polygonal. They are not irregular, they have a definite shape. They possess cell walls, distinct cell walls with primary and secondary layers, which contains substances such as lignin, pectin, tannins, and cellulose that makes them rigid, that makes their cell wall strong. Take your plant for instance. Some plants are woody plants. You see how strong they are. That's because they possess lignin that makes them strong, woody. Others have cellulose. These are components that make those plant cells very strong and solid, their cell walls. Whereas and then they also have within the cytoplasm, they have organelles such as your endoplasmic reticulum, Golgi apparatus, mitochondria, ribosomes, vacuoles, chloroplast, and nucleus. That is that about the plant cells. Let's look at the animal cells. Unlike what you have in the plant cells, the animal cells may be spherical, rectangular, cuboidal, spindle, or irregular. Unlike the plant cells that say they have a rigid structure, they have a regular structure and fixed shape. They are either rectangular, they are polygonal, or they are cylindrical, or thereabout. Animal cells are irregular. They can be circular, spherical, cuboidal, so they can just take any shape. They can take on any shape. They are very much irregular. They do not have a definite shape as you have in the plant cells. Two, they are much smaller than the plant cells. Animal cells are smaller in size. In terms of their size, the animal cells are much, much smaller than the plant cells. See? So you can begin to compare notes between the animal cells and the plant cells. The animal cells are much smaller compared to the plant cells. They are irregular, which means they can assume any shape, unlike the plant cells that are fixed in shape. Three, a typical animal cell is bounded by plasma membrane, which encloses the cytoplasm. They are bound by plasma membrane. They do not have cell walls. So the animal cells have plasma membrane enclosing the cytoplasm. That is the wall that is external, that is bound or surrounds the internal organs. Unlike in plant cells, where you have both the plasma membrane and the cell wall that has different components that make them rigid and strong. You see, but the animal cells do not possess such cell walls, but they are bound. They have a cell membrane or the plasma membrane that encloses the cytoplasm or the protoplasm. So the plasma membrane in the animal cell are semi-permeable. When you say permeability, it means access or inflow of material in and out of the cell. So if they are semi-permeable, means that they allow only certain solutes to pass through the cell, in and out of it. So that's the way they exchange substances or waste or materials, food items or nutrients in and out of their cell. They allow certain solutes to be able to pass through or in and out of their cell. So they are semi-permeable, And then finally, the cytoplasm contains food granules, Golgi body, mitochondria, central nucleus, which is centrally located, and other inclusion bodies. The cytoplasm contains organelles such as your food granules, Golgi body, mitochondria, centrals, nucleus, which are centrally located, and then other bodies, which we call the inclusion bodies, such as your storage vacuoles, where they store food, carbohydrates, and other um, substances. That is just about the animal cells. Now let's look at differences between the plant cells and the animal cells. Just as we looked at the differences between the prokaryotes and the eukaryotes, or the prokaryotic cells and eukaryotic cells. Let's see differences between the plant cells and the animal cells. In fact, even without spelling them out, we already can differentiate them now. We talked about their size, in terms of their shape, in terms of their cell wall, in terms of the components or the compositions and all that and all that. You can be able to now make out the differences that exist between the plant cells and the animal cells. But let's just take them out as we go. For the plant cells, one is that cellulose cell wall present external to the cell membrane. We said earlier on, their cell wall. The plant cells possess cellulose cell wall, which is external to their cell membrane. So after the cell membrane, you see the cellulose cell wall in the plant cells. You have the cytoplasm, you have the cell membrane or the plasma membrane, and then external to the plasma membrane, you have the cell wall, which possesses cellulose for the plant cells. Whereas for the animal cells, they had no cell wall. The outermost structure is the cell membrane or the plasma membrane. Animal cells do not possess cell wall. Plant cells possess cell wall, which has or contains cellulose. So the external structure for the animal cell is the plasma membrane or the cell membrane. Whereas the most external structure, the outermost structure in the plant cell is the cell wall. Take note of that. For the plant cell, the most external structure in the plant cell is the cellulose cell wall. Whereas for the animal cell is the plasma membrane or the cell membrane, which means that the animal cells do not possess a cell wall. Two, in the plant cell, vacuoles are usually large. And you see those vacuoles, these are spaces where they store things, either food, waste, or thereabout. Their vacuoles, their storage organs within the cells are usually large. You could see those large structures in the plant cells, which are called vacuoles. They are usually large. Whereas in the animal cells, the vacuoles are absent. And if they are present, they are usually very small. If they are present, they are usually very small, but mostly they are not present in animal cells. They do not possess vacuoles. But when they have them, they are very small structures within the cytoplasm. Unlike the animal cell, they are large structures. You could actually point them out in the cytoplasm. Number three, the plant cells, plastids are present, whereas for animal cells, plastids are what? Absent. They have plastids. You talk about plasmids and other extra chromosomal materials within the cell that also have a genetic makeup and can give them extra characteristics or features. We're not saying that plastids are present in plant cells, but they are absent in what? Animal cells. Number four, for plant cells, Golgi body are present in the form of units known as diatosomes. The Golgi body, they are present in form of units known as diatosomes. Whereas the animal cells, Golgi body are well developed, having two cysteines. For their Golgi body, the both of them, they all have Golgi body. But the differences that exist between the two of them is that for plant cells, the Golgi body present in the form of units called the diatosomes, while for animal cells, they are developed into two cysteines. Number five, for plant cells, centroles are absent, whereas for animal cells, centroles are present. Centroles are found in their chromosome. They actually attach them to the cell membrane, and then they actually decide where cell divisions begin from. They initiate cell divisions in the cells. I'm not saying that in plant cells, centroles are absent, whereas in the animal cells, the centroles are what? Present. Number 6, in plant cells, they have definite shape, like we said before. They have a definite shape. They could be rectangular, cylindrical, or polygonal, and not, irregular. Whereas the animal cells, their shapes are irregular. They could take on any shape, any form. That's a major difference between the plant cells and the animal cells. And then number 7, plant cells stores carbohydrate as starch, while animal cells they store carbohydrate as glycogen. Plant cells they store carbohydrates as what? Starch. While the animal cells they store their carbohydrates as glycogen. So that is difference between these are differences that exist between the plant cells and the animal cells. Thank you very much for listening, and this is where we're going to end the class for today. In our next class, we'll be looking at other aspects of it, where we look at the cell structure and their functions. Yeah, we look at the different components of the cell and what functions that they play. We look at the nucleus, the cell wall, the cell membrane, the Golgi body, mitochondria, ribosomes, and then we'll see what are their functions or what are their roles within the cell. That's what we'll be looking at when we come back in the next class. But as an assignment, I want us to draw the diagram of the plant cell and the animal cell. That will be your assignment for today. Make a well-labeled diagram of the plant cell and the animal cell. That is the assignment for today's class. A well-labeled diagram of the plant cell and the animal cell. So as a summary, in today's class, we talked about the cell. We defined what the cell is. We said the cell is the basic unit of the organization of life. It is a unit of protoplasm that is bound by cell membrane and possesses or contains a nucleus. It has 3 components: the plasma membrane, the cytoplasm, and the DNA. There are basically, the cell theory as proposed are made up of 3 statements. One, that all organisms are composed of cells, that cell is the structural functional unit of life, and all cells come from previously existing cells. Saying that there are basically 2 types of cells, the prokaryotic cells and the eukaryotic cells. The prokaryotic cells are made up of cells without membrane-bound nucleus and other membrane-bound organelles, while the eukaryotic cells are made up of well-defined nucleus with a membrane-bound nucleus and other membrane-bound organelles. We look at the differences between the eukaryotic and prokaryotic cells, and then we discuss the structure of the plant cells and the animal cells. And we say that plant cells, they have fixed shape. It could be rectangular, polygonal, or cylindrical. They possess a distinct cell wall, and then they contain organelles such as the cytoplasm, endoplasmic reticulum, Golgi body, chloroplast, and so on. While the animal cells are basically irregular, they can assume any shape. They are much smaller. The external walls are made up of plasma membranes, unlike the plant cells. And then the plasma membrane, they are semi-permeable and allows the movement of only solutes in and out of the cell. And then we look at the differences that exist between the plant cells and the animal cells, and then we've concluded for this class. And like I said, the assignment for this class is to draw. Okay, the assignment will be submitted. Yeah, when you come back, because I wouldn't say the next class because I don't know when you will be back, but it should be drawn a well-labeled diagram of the plant cell and the animal cell. Thank you very much. I'll see you in the next class. Yes. All right. The assignment will be on the foolscap sheet, please. On the foolscap sheet. Yeah, thank you. I'll see you in the next class.